Hello and welcome, dear students. Today we are going to discuss about the topic genotype and phenotype polymorphisms. The main objectives of today's lecture are to know about the basics of genotype and phenotype, to know about the phenotype and genetic variation, to understand the genotype dominance, to understand genotype-phenotype distinction, to know about the genotype and phenotype polymorphisms. Dear students, let's start with the introduction first. The fundamental question in biology is what makes a man look like a man? Even though humans belong to the same species, why are individuals looking different? If the genes are hereditary, then why do not children exactly resemble their parents? The answer to all these questions lies in the concept called genotype, phenotype, and their interactions towards the environment. The genotype is a set of genes in DNA responsible for unique traits or characteristics, while the phenotype is the physical appearance or characteristic of an organism. The idea of phenotype, which corresponds to the observable attributes of an individual, was coined in opposition to the genotype, the inherited material transmitted by gametes. In 1911, Wilhelm Johnson proposed that genotypes and phenotypes form two fundamentally different levels of biological abstraction. Since then, the challenge has been to understand how they articulate each other and how genotypes map onto phenotypes. Dear students, let's now understand the genetic polymorphism. Genetic polymorphisms are the simultaneous occurrence of two or more discontinuous genotypes or alleles in a population, also known as genetic polymorphisms. Genetic polymorphisms determine the diversity of individuals. They constitute the variation in the DNA sequences occurring in the person or population. Genetic polymorphism also determines the diversity of human beings. The predominant current day meaning of genotype is some relevant part of the DNA passed to the organism by its parents. The phenotype is the physical and behavioral traits of the organism, for example, size and shape, metabolic activities, and patterns of movement. Polymorphisms and mutations arise through similar biochemical processes, but the use of the word polymorphism avoids implying that any particular allele is more normal or abnormal. For example, a change in a person's DNA sequence that leads to a disease such as cancer is appropriately called a mutation, but a difference in DNA sequences that explains whether a person has red hair rather than brown or black hair is an example of polymorphism. Most of the genetic polymorphisms are unable to alter the gene expression and hence do not alter the structure and function of protein molecule. These genetic polymorphisms are considered silent. The polymorphic gene can alter the amino acid in the protein without any change in the functional capability of protein, while other variant of gene can change the gene expression, leading to the formation of variant protein with impaired function. Such genetic polymorphism is considered as pathogenic. This variant protein possesses either impaired enzymatic ability or complete absence of catalytic property, leading to failure of metabolic and biochemical reactions in tissues. The analysis of gene polymorphism has helped the genetics and clinicians to investigate the molecular basis of pathogenesis of diseases. The genetic testing and interventional gene therapy are the emerging fields of medical science which can mitigate the sufferings and disease burden of humankind. When we see the great diversity of human ethnicities, really we find it shocking that all of these different ethnicities share a genetically identical sequence at 99%. The range of their variants is only within limits of 0.1% of genetic sequences that differs between double chromosomal threads. It is a small ratio of variance, that is 1% indeed, but it is accountable for the multiplicity in persons' phenotypes. Generally, genetic polymorphism can be available in numerous designs comprising single nucleotide polymorphisms or SNPs, tandem repeat polymorphisms or TRPs, which include a variable number of tandem repeats VNTRs and short tandem repeats STRs, insertion deletion polymorphisms, transposable elements or ALU repeats, also known as jumping genes, structural alterations, and copy number variations or CNV. Dear students, let's now discuss the genotype. A genotype is the genetic content of an organism. It is the collection of genes of an individual. The genetic information is coded in the form of genes. In 1903, Danish botanist, plant physiologist, and genetist Wilhelm Johnson coined the term genotype. A genotype is a factor that determines the phenotype of the individual. It is the genetic makeup of an organism. The term can refer to the alleles or variant forms of a gene an organism carries that. Any diploid organism, for example, humans, have two alleles at each genetic position or locus, with one allele inherited from each parent, that is, one from mother and one from father, because humans are diploid. Each pair of alleles represents the genotype of a specific gene. If two alleles are identical, then the genotypic condition is called homozygous, and if two alleles are different, then the genotypic condition is called heterozygous. For example, tall character is represented by two alleles. If both are identical, then it is TT, that is capital T, capital D, and we call the condition as homozygous. If both are non-identical, then it is capital T, small t, and we call the condition as heterozygous. Wherein both the conditions constitute the tallness, alleles of the same gene are either autosomal dominant or recessive. An autosomal dominant allele will always be preferentially expressed over a recessive allele. Genotyping is the process that determines the genetic composition. Genetic polymorphisms are the simultaneous occurrence of two or more discontinuous genotypes or alleles in a population, also known as genetic polymorphism. Genetic polymorphisms determine the diversity of individuals. The following are some of the methods to determine the alleles and genotypes. Punnett square is one of the best and easiest ways to determine the genotype of an individual. This is like a mini chart developed by Punnett and is used to determine the potential genotype for an offspring concerning a particular trait or character. Polymerase chain reaction or PCR, developed during the 1980s, generates a specific strand of DNA using the template strand. Both are identical and represent the homozygous genotype. During the process, it is possible to determine the original quantity of the target sequence in the sample. A hybridization probe is used to determine physical characteristics because of the genotype. The process begins with a complete restriction digestion of the DNA. It runs on the gel and transferred to a filter membrane. Then the labeled probe is added to the filter and allowed to bind to a target sequence. Direct DNA sequencing is used to sequence the complete genome of an individual, which provides the individual genotype, the DNA sequence of that individual. Dear students, let's now discuss the phenotype. The phenotype is defined as the external appearance of an organism. Organism's morphology, physical appearance, structure, developmental processes, and behavior are the phenotype of an individual. It merely refers to the observable trait. This phenotype is dependent on two factors. One is on its genetic content and the other is on its environment. Both factors may interact, further affecting phenotype. Polymorphic is a condition where two or more different phenotypes exist in the same population of the species. The phenotype may continuously change throughout an individual's life. Along with the environment change, the morphological and physiological change associated with aging. Different environments can influence the development of inherited traits. A population is polymorphic when its members fall into two or more categories referred to as alternative phenotypes. There are many kinds of phenotypic polymorphisms with specialization in reproduction, feeding, dispersal, or protection from predators. An individual's phenotype might be randomly assigned during development, genetically determined, or set by environmental cues. These three possibilities respond to a mixed strategy of development, a genetic polymorphism, and a conditional strategy. Variation in the phenotype is required for the process of evolution by natural selection. Natural selection acts on the genetic structure of a population indirectly through the contribution of phenotypes. Genotype, along with the environment, gives the phenotype. The phenotype varies according to the environment. Dear students, let's now discuss the phenotype and genetic variation. Phenotypes can be influenced by genetic variation. The change in a gene of an organism in a given population are known as genetic variation. These changes are generally due to a mutation in DNA. Mutations are changes in the gene sequence on DNA. Sometimes the difference in the gene sequence can change the phenotype expressed in inherited alleles. Gene flow also contributes to genetic variation. New genes are introduced when a new organism makes its entry into a population. This new organism's entry introduces the new alleles into the gene pool that makes new gene combinations. New genes are produced due to the recombination of homologous chromosomes called crossing over during meiosis. This recombination of genes can produce new phenotypes in a population. That's why the crossing over is called as raw material for the evolution to act. Dear students, let's now discuss genotype dominance. The concept of dominance was the most significant contribution of Mendel in the field of heredity. He observed that heterozygote offspring could show the same phenotype as the homozygous parent, concluding that some traits dominated over other inherited traits. The following are some of the examples. First one, the complete versus partial dominance. Phenotypes derived from the organism's genes can be affected by dominance, but it does not change how the genes are inherited. Complete dominance occurs when the heterozygous phenotype is indistinguishable from that of the homozygous parent. Sometimes the heterozygotes display a phenotype that is an intermediate between the phenotypes of both homozygous parents. This intermediate phenotype is a partial or incomplete dominance. When this occurs, a range of phenotypes is generally observed among the offspring. Therefore, the offspring may show a variety of phenotypes. Each one will lie along a continuum bracketed by the homozygous parental phenotypes. Dear students, let's discuss the codominance. This occurs when the phenotypes of both parents are simultaneously expressed in the same offspring. Alleles from each homozygous parent combine in the offspring, and the offspring simultaneously demonstrates both phenotypes. Mn blood group in humans is an excellent example of codominance. An individual with the Mn blood group has both M and N proteins at the same time. Dear students, let's now discuss the overdominance. Sometimes offspring produces a phenotype that is not related to both of the parents. This is known as overdominance that occurs when a heterozygote has a more extreme phenotype than that of either of the parents, that is mother and father. This is also called gain of mutation. Dear students, the another one is multiple allelism. When we talk about multiple allelism, more than two alternative forms of a gene occupy the same loci in a pair of homologous chromosome in a population is called multiple alleles. The process of determination of a trait by more than two alleles is called multiple allelism. Multiple alleles occupy the same locus on homologous chromosomes. They control the same character, but the rate of manifestation is different. When two multiple mutant alleles are crossed to produce an offspring, the hybrid's phenotype is a mutant type but not the wild type. Human blood groups identified by Carl Landsteiner in 1925 are an excellent example of multiple allelism. Multiple alleles exist in a population where there are so many variations in a gene. It comes under a non-Mendelian inheritance pattern. The human blood group is made up of type A, that is IA, type B, IB, or type O allele, that is small I. Type A, that is IA, and type B, IB are dominant over O and are codominant. When they are crossed, the resulting genotypes makes either type A, type B, type AB, or type O blood group. Dear students, let's now discuss genotype-phenotype distinction. In 1909, Willem Johnson first proposed the difference between phenotype and genotype in the study of heredity when he was working in Denmark. The distinction in their genotypes and how those dispositions manifest themselves in the physical characteristics of those organisms, that is their phenotypes. He derived this distinction while working with plants and it influenced the pure line of heredity. It is used to characterize the relationship between studies of development, genetics, and evolution. He began his experiments in 1892. He bred barley and bean plants that had been self-fertilized, and he measured the physical dimensions of the seeds in every generation. Based on the external appearance and characters of the plant produced, he could distinguish them into pure lines. He demonstrated that the group to which a plant belonged was a more reliable predictor of the characteristics of the seeds produced than were the characteristics of its mother plant by applying the combination of pedigree analysis and statistical analysis. He contrasted the phenotype of a group or organism with its genotype, defining the latter as the individual qualities of those organisms. These experiments were mainly conducted to identify hereditary characters. The genotype-phenotype distinction was called as the transmission conception of heredity. Based on this, the characteristics of individual organisms are transmitted directly to their offsprings. One of the best examples of this is Darwin's pangenesis theory, in which the changing organs and tissues of an organism continually modify its germinal material, thereby impressing the characters of that organism on its eventual offspring. However, genotype-phenotype distinction said that the phenotypic characteristics of a mother and her offspring would arise under the influence of the same genetic material and passed from generation to generation and immune to the environmental circumstances in which it was expressed. Johnson's genotype-phenotype distinction explicitly prescribed a relationship between the study of development and the study of heredity. According to him, the genotype of an organism gives rise to the organism's phenotype during development under the influence of the environment. The distinction between genotype and phenotype is important in evolutionary theory, where the survival and mating of organisms depends on their traits, but it is the DNA held to be unaffected by the development of the traits over the life course that is transmitted to the next generation. Dear students, let's now discuss the genome growth and the evolution of the genotype-phenotype map. The genotype is a set of genes in DNA responsible for unique traits or characteristics, while the phenotype is the physical appearance or characteristics of an organism. The evolution of new genes is distinct from evolution through allelic substitution in that new genes bring with them new degrees of freedom for genetic variability. Selection in the evolution of a new gene can therefore act to sculpt the dimensions of variability in the genome. This constructional selection effect is an evolutionary mechanism in addition to genetic modifications that can affect the variational properties of the genome and its evolutionability. One consequence is a form of genic selection. Genes with large potential for generating new useful genes, when duplicated, ought to proliferate in the genome, rendering it ever more capable of generating adaptive variants. A second consequence is that alleles of new genes whose creation produced a selective advantage may be more likely to also produce a selective advantage, provided that gene creation and allelic variation have correlated phenotypic effects. A fitness distribution model is analyzed which demonstrates these two effects quantitatively. These are effects that selection the nature of the genotype-phenotype map. New genes that perturb numerous functions under stabilizing selection, that's with high pleiotropy, are unlikely to be advantageous. Therefore, genes coming into the genome ought to exhibit low pleiotropy during their creation. If subsequent offspring genes also have low pleiotropy, then genic selection can occur. If subsequent allelic variation also has low pleiotropy, then that too should have a higher chance of not being deleterious. The effects on pleiotropy are illustrated with two model genotype-phenotype maps: Wagner's linear quantitative genetic model with Gaussian selection and Koopman's NK adaptive landscape model. Constructional selection is compared with other processes and ideas about the evolution of constraints, evolvability, and the genotype-phenotype map. Empirical phenomena such as dissociability in development, morphological integration, and exon shifting are discussed in the context of this evolutionary process. Dear students, to conclude, genotype and phenotype are the two essential aspects of an individual's inheritance. Understanding these two independently and together is fundamental to studying, learning, and applying genetics in all organisms and populations. The genotype-phenotype map is the concept underpinning the classical concept of pleiotropy, polygyny, epistasis, constraints, and gradualness. Genetic polymorphism, whether studied in the form of allozymes, RFLPs, mini and microsatellites, variation, or DNA sequence, have become useful tools in a variety of research fields such as population genetics, evolutionary genetics, systematics and molecular phylogeny, human genetics, agricultural genetics, and forensics. Genetic polymorphisms through multiple alleles at individual loci or position provide a mechanism to tag a gene or a piece of DNA, providing a powerful tool for a variety of investigations. These investigations cover a wide berth of objectives, including identification of genotypes in paternity and forensic studies, tracking the movement of individuals in field studies following the progression of selection experiments in laboratory populations, mapping of quantitative loci affecting economical traits in plants and animals, mapping of disease genes in humans, and evolutionary comparisons of DNA sequences and chromosome organization between related species. The use of genetic polymorphisms are almost endless. Dear students, it was all about today's lecture regarding Introduction to Genotype and Phenotype Polymorphism. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.